Hello, students. My name is Professor Sims, and today we'll be talking about one of my favorite topics, which is DNA replications. Now, this didn't come easy for me when I was learning it, so I'm going to try to give you guys my best understanding of the basics of it because that's how I learned it, and that's how I'm going to teach you guys. Now, it can be a hard subject, but it can be fun. So let's get into it. What is DNA? That's what we need to start at first. DNA, which the word itself is really hard to describe or really hard to pronounce. So let's go to the dictionary word for what it is. DNA is basically called deoxyribonucleic acid, which is a complex molecule that contains all the information necessary to build and maintain organisms. So things like me and you, animals, everything, plants, everything has DNA in it. So it's a good understanding of it. Now, who created it? That's the good part. Who created DNA replication? It was created by two men, Matthew Millison and Franklin Stiles, which was published in 1958 in the PNSA. And they used a procedure that was called the equilibrium density gradient centrifugation. Now, basically, it was experiments demonstrating the DNA replicated semi-conservatively. And that DNA molecule serves as a template for synthesis of a new complementary strand. Pretty much, that's what it is. Now, describing it, it comes like this. DNA is made up of three building blocks: a phosphate, a sugar group, and four types of nitrogen bases: adenine, thymine, guanine, and cytosine. These are the four bases. As you can see in the description, these how they are made up. Now that is a perfect DNA. It looks like a braid to me, like some, braided hair, or you take some noodles and twist it up. That's what it looks like to me. But that's not DNA replication. That's just a DNA strand. This is DNA replication. You have an enzyme, which is called a heliocase, that splits down the middle, and that's how you unzip it. You got to unzip it first. Why is it replicated? Well, DNA replication needs to occur because existing cells divide to produce new cells. And it happens in the S phase. It's called the S phase, the synthesis phase of the cell before mitosis and cell division. And this is another example. More closer, you can see the base, you can see the four nitrogen bases, and you can see it unzipping. This is the best understanding of it. And this is the new DNA replication. You can see the two daughter cells, which is creating the new DNA. Two new DNAs.